diploid cell up here, and again, ignore this, okay? You would have an egg and a sperm cell up in the ovary, okay? Where does oogenesis occur? So you know it's in the ovary, but where within the ovary? What structure? The follicle, okay? Remember, each follicle, a multi-cell structure, which is single egg cell called an oocyte, develops, but not at the same rate. We saw that when that follicle is ripe and ruptures and expels the egg at ovulation in the body cavity, that egg is not mature yet, like the sperm. When it leaves the testes, immature, so is the egg when it leaves the ovary, okay? In other words, it hasn't completed meiosis II. It's arrested in development. And what stimulates it to finish meiosis II and become a mature egg cell? What has to happen? Here's waiting. Okay, it's waiting for the sperm. What has to happen? The sperm has to get into the egg cell cytoplasm to stimulate it to finish meiosis II and become mature. Until that happens, it's just hanging out there in the oviduct and starting to pass slowly down. The sperm does not read it and enter the egg cell cytoplasm. It degenerates and passes out the menstrual flow. It never matures. Okay? It only is matured when a sperm gets into the cytoplasm. That stimulates it then to complete meiosis II and become a mature egg. Until that, it's not, and it won't mature. Okay? And again, fertilization in humans usually occurs in about the upper third of the oviduct, so fallopian tubes. That's why the sperm has to be motile. Okay? It's not motile, it's not going to make it. The egg is waiting for it as it comes down. Okay? And then actually we're going to see that embryonic development actually begins when fertilization occurs in that oviduct, according to Gitzner's implants. Okay, so here we are, starting with again a diploid body cell. This is called the oogonium, right here. It too undergoes mitosis, not meiosis. Here's another cell like itself, okay? Another daughter cell is going to be the primary oocyte. That's what this cell is, primary oocyte. It too is diploid. Remember, mitosis. Mitosis, you don't change your chromosome number, you start with a diploid cell, you end with diploid cells, okay? So what is going to be this primary oocyte? Another one's going to be basically a cell that's going to be another oogonium, okay? This cell then, a primary oocyte, undergoes meiosis I. It produces two daughter cells. One is going to be a secondary oocyte, haploid. Other one's going to be just a little teeny cell that's not to scale here, called a polar body. Now, the polar body is just a means of getting rid of excess chromosomes. You don't want equal division, cytokinesis, because the cytoplasm is precious. And the egg is going to contribute most of the cytoplasm to the embryo. That's why we have diseases that involve organelles like the mitochondria and mitochondrial DNA. They're rare, but often very dangerous diseases. It's always the maternal line. Be we male or female, we got auto organelles originally derived from our mother's egg cell. All the mitochondria, the endoplasmic reticulum, Golgi, and everything came from the egg cell, not the sperm. The sperm, like I said, just a tiny bit of cytoplasm. It's got mitochondria to power the flagellum, and negative digestive enzymes in the nucleus. That's about it. Like I say, the sperm is only there to get those chromosomes, male chromosomes, into the egg. Everything else better be there already, because the male's not going to contribute anything. Okay? Just the chromosomes. Egg's got to do the work. All right? What's new? Okay? It's like life, right? One does all the work. Okay. So we got two cells here. They're both haploid, a secondary oocyte, and a polar body. Again, this is not to scale. Now this cell then is going to undergo meiosis II. If it completes meiosis II, it'll become a mature ovum. But it's only going to do that if a sperm cell gets into its cytoplasm. When it leaves the ovary at ovulation, it's in this state, okay? It's a secondary oocyte. It hasn't completed meiosis II. The polar body may divide again and produce more polar bodies. Doesn't matter, because eventually they're all going to degenerate. As I said, the polar bodies are just there, get rid of excess chromosomes without wasting a bunch of cytoplasm. That's why these things are tiny. You don't want to waste precious cytoplasm when these cells are going to degenerate. The precious cell is the oocyte, which can become the ovum. Like I say, it's got to have everything there. Just waiting for the sperm, okay, to wake it up and say, finish, okay? Let's get this process going. With all the organelles again, cytoplasm of the egg. The only thing a male is really contributing is male chromosomes. down here, and it's not quite accurate, there should be a sperm cell here. This is the mature ovum. It should only be if the sperm cell is already in the egg cell cytoplasm, and then, of course, you get the diploid cell, which is the zygote, the fertilized egg. Okay? And notice, we started with one precursor cell, the oogoid, diploid, and how many eggs did we end up with? One. One for one. That's because these extra cells, the porter bodies, are going to degenerate. They're not important. Okay? We only want one good egg and forget all these porter bodies. Like I say, porter bodies are just a way to get rid of these excess chromosomes and not waste cytoplasm in the process. Okay? Any questions? Again, where does this occur? Oogenesis? Within the follicle, inside the eggs. Again, I would like to know the cells here by name, whether haploid or diploid, and if they're human cells, how many chromosomes. The diploid human cell, 46, 23 pairs. Haploid human cell, 23 single chromosomes, unpaired. Okay? So we studied these charts. Now I'm going to start with the label charts, and then you get confident for the unlabeled